Okay, hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Paula Foster. Dr. Foster is a professor in the Department of Medical Biophysics at Western University and leads the Cellular and Molecular Imaging Research Program at Robarts Research Institute. The research is mainly focused on the development and application of MRI cell tracking techniques and iron and fluorine-based cell labeling technologies for in vivo imaging of cells and basically in experimental animal models. A major focus of her research, where she had made significant contribution and achievements, is tracking cancer cell dormancy and metastasis and inflammatory cells associated with tumors in preclinical model of breast cancer and melanoma, and also prostate cancer. Cellular therapy is another exciting area where these cell tracking techniques may also be super important. More recently, the Foster Lab has been studying magnetic particle imaging, MPI, for in vivo cell tracking. I would just like to remind the audience to please address any question you have using the Q&A box or raise your virtual hand. Dr. Foster, thank you so much for coming here today. The virtual stage is all yours.
Thank you very much. I appreciate the invitation to speak today about my research. So just the agenda for today, I'm going to start by telling you about the clinical problem, which is going to be brain metastases due to breast cancer. And then I'm going to go on to talk about cell tracking with MRI and how it's used to characterize the fate of metastatic cancer cells in the brain. Then I'm going to talk a little bit about MRI and BLI or bioluminescence. And finally, I'll talk about MRI and MPI or magnetic particle imaging. And I will spend a little bit of time talking about how MPI works since it's such a new technology. So the clinical problem. Breast cancer is now recognized as the world's most commonly diagnosed cancer, with 1 in 8 women in North America developing breast cancer at some point in their lives. Unfortunately, I'm sure everyone in the audience knows about metastases. Cells can actually break off from the primary breast tumor and extravasate, entering the bloodstream or the lymphatics, and then traveling to a distant organ where they could form tumors. And this is known as metastasis. So with breast cancer, up to 30% of breast cancer cases will metastasize to other locations. And these locations are most commonly the brain, which is what we're going to talk about today, but also the lungs, bone, and liver. And brain metastases in particular has a devastating prognosis. So there's no cure. Mean 1-year survival is only about 20%, and 2-year survival is less than 2%. So really a devastating diagnosis. To understand metastasis and its underlying mechanisms, it's important to be able to track the fate of metastatic cancer cells. And there are essentially 3 fates of these cells. The first being apoptosis. So after the cells left the primary tumor, many of these cells will just die and they'll be cleared by immune cells. The second fate is proliferation. So these cells can extravasate from the blood vasculature, enter another tissue, and then continue to divide and then form macro or micro metastases. And lastly, the third fate is dormancy. So cells can extravasate and reach another tissue, but then remain dormant. So this is where they are still viable, still live, but they're not proliferating. And it's thought that this population of cells is what contributes to recurrence. They also present a substantial therapeutic problem because they don't respond to therapies that target proliferating cells, such as many chemotherapies and radiotherapy. So what exactly is cancer dormancy and how is it described in the literature? Well, there are three sort of categories: tumor growth, tumor dormancy, and cell dormancy. We are going to be focusing on cell dormancy, and this is, as I said, the condition where the growth and the death of the cells is balanced. So I'm going to now move on to talk about the imaging methods that we have used to study this problem. So first of all, cell tracking with MRI and iron nanoparticles. Our lab's been doing this for quite a long time and have developed some technology and some methods to track cells this way. So just as an introduction, there are a variety of different kinds of superparamagnetic iron oxide particles that people use for MRI cell tracking. There are a range of sizes, as you can see in this schematic here. Mostly this variety, so mostly 50 to 100 or say 200 nanometers. And these are dextran coated and biodegradable. There are some smaller ones that people would use more for intravenous administration and in situ labeling. And there are some larger ones, which we call MPIO, micron-sized or microspheres. And a lot of the work I'm showing today is using these MPIO. And these are typically like a polystyrene bead with iron cores embedded in it. And some of these can be purchased with fluorescent tags as well. On the right, you can see some of the images of cells that are labeled. So first a GFP positive cell, so it's already green, and some red fluorescent particles. In the center, electron microscopy of a MPIO-labeled cell. And on the right, Pearl's Prussian blue staining for iron. So those are sort of typical microscopy methods to look at the iron in the cells. The iron's typically taken up in vitro in culture overnight and compartmentalized in the cells within lysosomes, as shown here. Okay, so why do we like iron for this? Well, iron has a very strong effect on the local magnetic field, causes inhomogeneity or dephasing, which leads to regions of signal void. And you can see that in the image below, which is of iron-labeled glioma cells in the mouse brain. This effect is much larger than the actual cell. The disruption to the field extends quite a ways, and that depends on how much iron there is and on your imaging parameters, but we refer to that as a blooming effect. And so it has very high sensitivity. This example here shows you an example of high sensitivity. So in this paper, iron-labeled cells were injected into the heart in mice, and they arrest in the brain. And in the image at the top, you can see these discrete regions of signal void. Sometimes they are small clusters of cells, sometimes two cells, sometimes single cells. And the example shown here is of a single cell in the brain and confirmed by confocal microscopy, where you can see the green from the cell and the red from the iron particles. So under some conditions, single cell detection is possible. So this is the model that I'm going to be talking about today for cell tracking in the mouse brain. So we're going to be using a human cell line, 231BR, which is brain metastatic. So these cells are going to be labeled with MPIO, and then they're injected into the left ventricle of the beating mouse heart using ultrasound guidance. And as I've said, they arrest in the brain. They arrest in other places as well, but they preferentially grow in the brain. So you only occasionally see some tumor growth, say in the liver or lung, but it's almost all in the brain. And then for our studies, imaging takes place over some time course. So this is what the mouse brain looks like on day 0, the day that we inject the cells. This is one animal, just some MR image single slices through different levels of the brain from a 3D data set. The front of the brain, of course, you can see the eyes, and the back of the brain, midbrain. And you can see individual voids throughout the brain. So these cells arrest everywhere just due to size restriction. And we can do things again, like look at the fluorescence associated with the cells or the particles to validate that. So after day 0, a couple different things happen relating to cell fate. Proliferative cancer cells, so those that are going to divide and form metastases, they gradually lose the iron signal through dilution. You can see that in the cartoon here where the cell is splitting and the iron particles are getting split as well to daughter cells. Eventually, there's not enough iron in the cells to see the void. And what takes over is contrast related to the changes in T2, which are related to accumulation of water with tumors. So you can see now, day 19, 21, 26, 29, we have tumors forming. We usually see them as early as 19, and tumors growing in the brain, and you see a hyperintense region now associated with that tumor. The other thing that can happen, though, is related to the non-proliferative cancer cells. So there are some cells that are not going to divide. Here, the cartoon kind of shows you that those cells will keep their iron. So they will retain the iron and will still be visible. And so those are the quiescent or even slowly cycling will, the voids will persist. And you can see that in these images. Here, day 0, 21, 29, you see many of the voids are persisting, but we also have some voids turning into tumors. This is sort of a bigger picture of a single mouse brain looked at over time. So it's the same image slice over time, focusing in on the 3 different regions. And just to show you the different things that are happening. So if you look at 3, you can see we're looking at this void here, which disappears for a while. So that presumably is a cell dividing, losing its label, and then in that area you get a tumor forming. Here you have a big cluster of the voids and a bigger tumor. And here you have 2 voids, one of which looks like it has a tumor forming around it at the end. So all this data can be put together to make these fancy movie versions of it, which here the voids are now colored red. So this gives you, an idea of the whole brain. You can see that again, as I said, the voids are everywhere, but most of them die and are gone somewhere between day 14 and 21 when we're imaging. And you can see that there are many fewer voids there now. And then with this sort of 3D data, you can then render where the tumors are, and you see those in green here. So you can see that there are a lot of cells that haven't lost their iron in this image. So with that data, we can create a cell fate map. And by doing this, we're comparing day 0 images with day 28 images. And we can sort of determine 3 different fates: the transient cells, which are the voids that disappear, the proliferating cells, which are those that form the tumors or the hyperintense regions, and the non-proliferating cells, which remain voids. And it's these ones that we believe are related to dormant cancer cells and that we're very interested in. So our working hypothesis has been that the retention of iron oxide particles in non-proliferative or slowly cycling cancer cells can be exploited to monitor the fate of this subpopulation in vivo with MRI. And so I'll show you a couple studies where we have tested this. One of the studies is using radiotherapy. So whole brain radiotherapy is a treatment for brain metastases in patients. And in Canada, it's used alone for primary treatment if there are more than 3 metastases in the brain. It's used as an adjuvant therapy with surgery or stereotactic radiotherapy to control microdisease. And this will be delivered after tumors are detected in the brain by MRI or CT. That's traditional. There's also something known as prophylactic treatment, where you would actually give the radiotherapy before any tumors have grown in the brain. And this is used as a preventative treatment for patients who are at high risk of developing brain mets. So it's used for small cell lung carcinoma patients. It's being considered in Canada for high risk metastatic breast cancer, such as HER2 positive. So in this study, we're using whole brain radiotherapy on mice, and we have a novel image-guided microradiation system at Robarts, which we've developed for mouse radiotherapy, and it allows us to precisely outline the mouse brain and isolate the radiotherapy, so we can move a little closer to mimicking the clinical situation. So pre-treatment images with CT are required, and you can see here from the calculated dose distribution, we can get down to half a brain. And here you see the DNA damage by gamma H2AX staining. But in this study, we're doing whole brain. So first, the traditional radiotherapy. So in this case, we are using the HER2 positive 2-1BR cells. And here we are giving 20 gray delivered in 2 fractions. And we chose that based on the biologically effective dose. And that's a calculation that's used in radiobiology to quantitatively compare the biological effect of different radiation treatments or fractionations. And so here we waited till day 23 when we knew we could see tumors in the brain, like what would be done in patients. And then we gave the radiotherapy in 2 fractions and imaged on day 32 and 36. And you can see here that this form of radiotherapy controlled tumor growth in the brain. So the treated animals are the white bars, and by the end point anyway, you can see that the tumors are being controlled there. And they also have a much smaller volume here, the images, MRI and histology from the treated and the untreated. So this traditional approach works well in this model. We also looked at this early whole brain radiotherapy or prophylactic approach. And so here, cells are injected again, that's day 0. And here we gave whole brain radiotherapy the next 2 days, so right away after the cells arrested in the brain. And then we imaged at day 4 and at day 32. You can see that this eliminated almost all metastasis formation in the brain because it's killing cells early. So you can see there's some small number. There were a couple of tumors and a couple of mice in the hindbrain, probably where the radiation did not get to. So what was interesting is shown here. So what we didn't see was any difference in the number of signal voids that persisted in the brain. So I showed you just before that we always have voids persisting throughout the experiment. Most of them die. And you can see here, so this plot shows the percentage of voids remaining at days 0, 4, and 32. And as usual, it declines over time. And the point is here, though, that it was not different for the 2 groups at any time point. So it indicates that metastatic cells in the brain do not respond uniformly to treatment and suggests that whole brain radiotherapy leaves non-proliferative cells untreated. And in fact, that is not surprising because we know that dormant cells are not responsive to these types of treatments. We were trying here to monitor mice that received prophylactic radiotherapy for a long time. Because they weren't developing tumors, we could do this. And we were hoping to be able to monitor iron retaining cells until they eventually formed tumors. So But out till 95 days, we were still only seeing the signal voids. So it's very hard to model recurrence from dormancy, especially because some human tumors can recur as long as 20 years later. So the next project I want to talk about focuses on something called concomitant tumor resistance, or CTR. And CTR is a phenomenon in which a primary tumor inhibits the growth of metastasis. So it's rare, but there is clinical evidence of CTR where surgical removal of tumors can be followed by an abrupt increase in metastatic growth. And this is very interesting and potentially very important thing to understand, but right now the mechanism is really not clear at all. So we developed a new model for CTR where we have one group where 50,000 breast cancer cells, 231, are implanted into the fourth mammary fat pad. So it's actually group 2. I have that opposite. And afterward, so this is to establish a primary tumor. And then later, day 15, MPIO-labeled 231BR cells are injected into the heart, as I've described before. So this is the parental cell line of the 231BR. And then in another group, we are there's no primary tumor, just the intracardiac injected cells. And so we're imaging to see, well, to monitor the primary tumor as well as the metastases that develop. And so this just shows the imaging timeline. So we're imaging on day 0 to see the arrest of the cells. And then we're waiting till day 30 to evaluate tumor growth and persistent signal voids. So what we found was that the mean number of brain metastases that developed by day 30 was significantly lower for the mice that had a mammary fat pad tumor. You can see that here. The mean tumor volume was not different, but the number of tumors was different. And so this indicates that the presence of the primary tumor is somehow suppressing the development of secondary metastases. So that's interesting. What was more interesting was that there were more of the persistent signal voids, or non-proliferative cells, in the brains of the mice with the primary tumor. So you can see that here. There's a significantly more of the signal voids present in these animals here, which are the ones with the primary tumor. So this suggests that the presence of the primary tumor also is enhancing cell dormancy, suggesting a possible novel mechanism for CTR, that the primary tumors restrict the growth of the brain metastases by inducing or maintaining dormancy somehow. So this is something that we are continuing to study in my lab. So let's just summarize the MRI quickly. So I showed you there's very high sensitivity for MRI-based cell tracking. Some other things to discuss, though, are relatively low specificity. So by that, I'm referring to the fact that I think you could see there were other black regions in the images, which could possibly complicate your interpretation of the voids that are of interest. And also, you can't differentiate between live and dead cells with MRI. So that leads to low specificity as well. The other thing is the indirect quantitation. So since the iron is not directly detected, it's detected indirectly through its relaxation effects on protons, it's not possible to really reliably quantify iron concentration. And so we can't get to cell number. On the plus side, there's no signal attenuation with iron-based MRI. So I'll talk just a little bit about BLI, or bioluminescence imaging, because we've started to use it quite a bit as a complementary imaging modality. And it's very commonly used to, for tumor studies. Just a quick review of how it works for those who don't know. You use luciferase expressing cells. So these can be engineered or purchased. And these cells are injected into mice to either form tumors or to have cancer cells arrest, as I've shown you. And then just before imaging, the substrate, the corresponding substrate has to be injected. And visible light is produced during the luciferase-mediated oxidation of that substrate. So ATP and or oxygen are required in this reaction. And so we're only detecting live cells. The CCD camera then spatially encodes the intensity of the photons and can generate an image, and you can also quantify the radiance. So we've used this in the same model I've been talking about. Here we looked at day 0, MRI and BLI. This image shows 2 image slices through the mouse brain, just as representative slices, and the signal voids that are present on day 0. And you can see here that in the same mouse, BLI signal was detectable in the brain where the cells arrest. The number of signal voids, total number of signal voids correlated with BLI signal, as did the percentage of black pixels. So 2 ways to sort of, 2 semi-quantitative ways to analyze the MRI data. So this data itself provides some evidence that the regions of signal void are related to live cells. We then looked at these mice over time. So that's what you're seeing here, the same mouse being tracked over time. And I guess this is day 0, day 8, 21, 28. So again, day 0, you can see voids in the MRI and BLI signal in the brain. Now, day 8, you see no signal with BLI and very little or no voids in the mice. And I showed you this earlier. There's a time point where these cells are both dying and dividing, and many of them fall below the detection threshold for MRI. They're also falling below the detection threshold for BLI. Here you can see new signal in the brain with BLI. So now the cells are proliferating, and this is indicating higher levels of live cells, and that continues over time. So this gets larger. You do also see in this animal some growth in the liver region. So these correlate really well. So quickly then, so BLI, I say moderate sensitivity. I mean, it depends on many things, the type of luciferase. There is a paper that shows single cell detection with BLI and Acknowledgments, but it depends on a variety of factors. It does have very high specificity because it's only detecting those luciferase positive cells and only those viable cells. However, it does have, again, indirect quantification. We can't get cell number really. And the other problem with BLI, which I'm sure many people understand, is the light scattering and depth limitations. So signal can be attenuated at depth. So I'm going to talk about MPI now. So this was first invented, I think, in 2001 and published in 2005 by Gleisch and Weisenecker at Philips. Canada has just one MPI system, and lucky for us, it's installed at Robarts. It is a momentum MPI that's from Magnetic Insight. There are 2 commercial preclinical scanners right now, one from Brooker and one from Magnetic Insight. And it detects superparamagnetic iron oxide. So you can guess why we wanted to explore this. So MPI is a hotspot or tracer imaging, it's referred to, because you are only detecting the iron. There's no contrast. And therefore it's also very, very specific. The reason I am pursuing MPI really is because of its quantitative nature. So the signal is proportional to iron. And so you can image a cell sample and a mouse, and you can get the amount of iron per cell, and then use the signal from the MPI image of the mouse, and you can actually calculate a cell number. I'm going to say medium sensitivity for this right now, although it's very early days. But in the literature, the lowest I think you're going to find is 1,000 cells in vitro and 2,500 cells in vivo, or down to 10 nanograms of iron, not in cells. It does require an anatomical image like other tracer imaging does. And so here you can see an example of a mouse with a focal region of iron injected, and also with a CT. So Magnetic Insight does now have an MPI system with a CT. Okay, so this image really illustrates nicely this issue with depth and the limitation there. Here are two regions of iron injected into a mouse. It's the same amount of iron in both regions, but they're at different depths. And so if you quantify this, you'll get the same amount of iron. If you look at the BLI signal, you can see that the signal is much lower from the deeper cell source. Now, there are some things you can do to remedy that. You could image both sides of the mice and so on, but that illustrates that problem. And then this is the MRI of the same mouse, where the issue of specificity is well illustrated. So this is a pocket of air, this is the pocket of iron, and they look pretty much exactly the same. So how does it work? I want to just spend a few minutes describing how this works, because many people probably haven't heard of it. It's built around a gradient magnet system. So there are two opposing electromagnets. You can see them here. This is inside the system and in the cartoon here. They face each other and they create a strong gradient magnetic field. And within that, there is what is called a field-free region, or an FFR. And this is where magnetic fields are beginning to cancel out. And so we call it a field-free region. It's a very, very low field here. This is the sensitive region in MPI, okay? So these are strong gradients, so 3 to 6 Tesla per meter versus MRI, which is in millitesla. And these are weak magnetic fields, so millitesla level instead of Tesla. So the MPI technique really relies on spatially selective saturation of particles. Here, the red arrows refer or represent the SPIO particles. And in a magnetic field, so in the gradient field, they're strongly magnetized and they saturate. In the field-free region, they have zero magnetization, okay? And this is superparamagnetism. So the characteristic where the magnetization can change when in or out of a field. And this is really key to MPI. So what happens is the field-free region is moved around to image the field of view. And as it moves around, it detects the response of the iron particles to the change in the magnetic field. And I'll show that with a couple of examples here. So here again, you can see that this is representing the iron particle. And you can see this change in magnetization that is represented by a Langevin function. And the MPI signal we see is the derivative of that, a point spread function. So that change in voltage, or that change in magnetization, induces a voltage which is measured with an RF coil. And it's proportional to iron. So one more sort of schematic here for you, just to show you. So this field-free region is being shifted. It's shifted along the x-axis by changes in the electric field strength. This is called the drive field. It's shifted along the y-axis by translating the sample bed through the scanner. And it scans in a grid formation, so left, right, bottom to top, to create a 2D image. And as you'll see in the next slide, to get a 3D image, it is shifted by shifting the whole gantry around. So hopefully that gives you a bit of a sense of how it works. The type of iron particle is very important for MPI. The core size, clustering of particles, the distribution of cores within the particle are all very, very important. In MRI, we talk about hydrodynamic size, and the size affects how much iron is going to be taken up by a cell or its biodistribution. But in MPI, it's all about the core size, the iron core size, and things like cores interacting with each other. And I don't really have time to go into much of that today. But the ideal SPIOs or iron particles for MPI are still under development. Right now, the best are in the range of 20 to 30 nanometers. And there's lots to say about that, but I also don't have time. The system has an MPI relaxometry mode, which allows you to characterize nanoparticles, which is nice. And you can look at the relaxation of these particles. So that flipping of these spins is also referred to as relaxation in MPI, just to make things confusing. And it's the lapse in flipping, because it's not really instantaneous. That is the relaxation mechanism, or the relaxation. So relaxometry looks at this, and we can look at sensitivity. So here you look at peak height. So here particle B is higher than particle A, so it has a higher sensitivity or higher peak signal. And we can look at resolution. So the resolution, we look at the full width at half max, and here you can see particle B has a narrower full width half max, and therefore better resolution. We've looked at the relaxometry of several commercial agents. So here we looked at fureucarbitran, which is actually Resivist, which is an MRI contrast agent, or was. And it's actually the most widely used right now for MPI. I think that it was just tested and it worked, and people have used it. So it's actually not at all ideal, but it has a bimodal distribution, of course. So some of them are good, some of them are in the right size, and some of them aren't. But we've been comparing things to it because it is the most widely used. We looked at MPIO because I've done all those previous MRI experiments with MPIO. We looked at Synomag-D, which is a commercially available tailored MPI particle, which is much better than the others, actually. And we looked at Freemoxtal, which is a USPIO that is basically used in humans now. It's also used for iron replacement therapy, and it's a small, very small particle. So you can see, looking at the values here, the relative sensitivity. So Synomag-D is about 2.4 times better, and has about the same resolution, close to the same as fureucarbitran. And you can see the sensitivity here, and the relaxation shown here. You can also just see it by eyeballing it. So Freemoxtal has very low resolution. You can see that's very blurry. MPIO surprised us, really. It has low resolution, but it has quite good signal. It produces a lot of signal. Remember, they have a lot of iron embedded in them. I don't know much about the core size because it's hard to get that information, but you know, it's something we have explored. So that's something you can do with relaxometry to characterize particles before you use them in vivo. And so one of the first or early things we did was to look at MPIO-labeled cells in the mouse brain, just like I've shown you with MRI. And here's the MR image. So this is a very nice image, high resolution. You can see voids. And then this is the MPI, so low resolution. And that is really the biggest limitation of MPI right now. And it's something that I think the whole field is really working on improving. But in this example, 50,000 cells were injected into the heart. We measured a sample of the cells and found that they contained, on average, 30 picograms of iron per cell. We know that 5 to 20% of the cells that are injected intracardiacally arrest in the brain. That's a wide range, but it's a difficult injection, and there's mouse variability. With these 6 mice, we measure between 0.2 and 0.7 micrograms of iron per brain. If you look at the higher numbers, say we got 10,000 cells in the brain, that would be 0.3 micrograms. So there are a number of factors that really make it impossible to calculate true cell number in this model. First, the measurement of iron per cell, that's an average value. Some cells will have more iron, some will have less. Again, the delivery to the brain, that is an estimation. These injections are technically challenging, and while we do have significant expertise, not every injection is going to deliver the same number of cells. But considering these caveats, this number, 0.3, actually is in good agreement with our measured values from the mouse brains. Now, the next thing we did in the mouse brain was to use Synomag-D. As I said, it was developed for MPI. It's monodispersed, multi-core, the right core size, dextra-encoded, has about twice the sensitivity, and I think it's more like, twice the resolution of MPIO. Yes, that's correct. And here are the images from these mice. So in this case, 250,000 cells were injected intracardiac, only 5 picograms of iron per cell. So MPIO was 30 because they contain so much iron. That's why we like them for MRI. Again, here we're going to get between 12,500 and 50,000 cells in the brain. And we measured an average of 0.08 micrograms here. So that turns out to be roughly 16,000 cells. So again, in the range. And so this work is continuing. You can see that these images are higher resolution, and that's the Synomag-D. We also looked, we've gone a little bit longitudinally with this, just out to day 7. What we have seen is a decrease in iron content over time, totally expected, because as I've shown you a couple times now, these cells are going to die, clear, and or proliferate and lose signal. However, we were able to measure signal at day 7 and see signal at day 7. If you remember, with the MPI, with the MRI and the BLI, we lost the ability to track cells around day 7, 8. And so at this point, this is just on average 10 nanograms of iron, which is the lowest we've detected. So finally, to summarize, so far I've shown you that MPI has moderate sensitivity, high specificity, direct quantification, and no signal attenuation. So it's not perfect. I have only listed 4 things here. Resolution is a big problem. You need anatomical images as well. So there's lots of work still to be done with MPI, but along with that, I think even better sensitivity and better resolution. And finally, to summarize the cancer imaging side of things. So in the clinic, the diagnosis with MRI is using gadolinium enhanced MRI. So non-proliferative cells can go undetected in the clinic, or do go undetected in the clinic by conventional imaging, and untreated by conventional treatments. So this is a picture in the clinic versus the picture that we see in the lab. So it's important to understand the rest of the cells that are in the brain. MRI, I've shown you can detect and monitor both proliferating and non-proliferating cancer cells in vivo over time, from their arrest to tumor formation. BLI will provide information about cell viability, and MPI allows us to quantify the cell number. So all of these things, and in a complementary fashion, really, will be useful for our studies of metastatic brain cancer. So I just want to acknowledge some of the people who contributed the research that I've been showing you, some who've helped develop the technology and sources of funding. Thanks very much.
Thank you very much, Dr. Foster, for this great talk. And we now move to the Q&A phase. I'd just like to remind the audience to please address any question you have using the Q&A box, the chat, or just raise your virtual hand. And while people are typing, I'll use the opportunity to ask one of my questions. I would like, if you could please comment on the current status of translation of SPIO imaging in humans. There was a big hype about it, and then I'm not sure where it stands with the ferromoxital status, or what are the current challenges?
Yeah. As far as translational stuff with MRI and iron labeling, I feel kind of old-fashioned, I'm going to say, because now we've seen papers recently that talk about CEST and not having to even label cells. So there's all of that. The ferromoxital is being used. There are a number of really good papers out in the last few years that have used that to image things in the brain, including a study that used it to label tumor-associated macrophages in situ in glioma in humans. So I think that's another important application. As far as MPI, there's no reason there can't be a human MPI, as far as I understand. There are groups building them. There are groups testing them. Magnetic Insight has funding to build one, and there's several other groups building them in their labs. And there's actually the World MPI meeting in 2 weeks, where I'm pretty sure we'll be seeing some human systems.
Awesome, thank you so much. And the next question from Bruce, can you please unmute?
Hey there, fantastic talk. First, I'll just mention for those listening from the center who may not realize it, we are one of the groups that are building human MPI systems.
Oh, okay.
So quite far along on that. We built our own rodent system. This is Larry Wald and Clarissa Zimmerman and others within the so-called PIGS, the Physics and Instrumentation Group within the center. And they're quite far along on their human system. I don't believe they'll have human data for this year's meeting, but we presume by next year's meeting, and we're very, very excited about it. So thank you for introducing this to us, from the outside. And I just wanted to let others in the center, if you're interested in learning more about MPI, of course, read all about it based on this talk, but also go talk to your colleagues right in Building 75. So that was that, great. But I'm curious, one technology you didn't talk about, albeit I showed up a little late, so I apologize if I missed it, but is PET. PET obviously has high sensitivity, potential specificity if you can, kind of tag appropriately. Spatial resolution somewhere between MPI and MR, I suppose, might be a fair way to characterize it. Where do you see the role of PET fitting into these kind of studies, and especially for human translational work?
Yeah, PET's huge, and I just don't do any PET, so I can't talk about it. But I think you said, I agree with everything you said. If I were going to put it on the slide and compared to the other modalities, yeah, it also has a lot of good features. And I know, you know, we're going to see a lot of interesting PET cell tracking at the European meeting, which is next week. And you know, it's always going to have a role in preclinical and clinical cell tracking. I think it's ahead of the game for everything really. And it's really cool to hear about your MPI work. I wish I could have came to.
Yeah, I'm sorry you're not here that we can show it to you ourselves, but I know our colleagues would be delighted to share with you what they're up to and learn from you, and especially start thinking about the applications that you've been pioneering. You know, the group has been focused very heavily so far on the technology, have made, I think, some really nice innovations that have really pushed the limits of sensitivity, et cetera. But now, of course, the question is, what are we going to do with the device? And you've given us some tremendous ideas, and I'm sure they'd be very open to collaboration should that be appropriate.
Yeah, that would be fantastic. And I'm actually planning to be back in Boston in the summer sometime. So.
Oh, well, please come and visit. Let us know when you're coming, and I know we'd love to show you what we're doing and talk about potential collaborative opportunities. That's great. And thank you again for the talk.
Yeah, absolutely. I'd love to do that, and I will do that for sure.
Good. I'll look forward to seeing you this summer then.
Yeah, next question from Peter, please.
Well, hi, Paula. I had a few questions. I guess like the first is, do you think, or is there anybody working on ways in which to make the particles sensitive to cell viability? Do you think there's any hope to be able to do that?
That's a great question, Peter. One thing that MPI is sensitive to is a change in between free iron and compartmentalized iron, just like with MRI as well. So if a cell were to die and lose the iron, and that was to change, that could be potentially detected. There are, I think, just 3 papers out there that I know of that compare or model intracellular and free, or bound and compartmentalized, that are looking into that. So, and with MRI, that, you know, that was also something that MRI was sensitive to, but it was never sensitive enough to be able to use it really preclinically to say for sure what was, you know, that something changed. So I think there's the potential there anyway.
No, thanks. I had another question, which was about labeling immune cells. And I know you talk about this, but I was wondering, you know, are there, or in cell types in general, like, I mean, are there different technologies that work better with certain cell types?
Different ways to label them, you mean?
Yeah, you know, like, yeah.
So with iron particles, we've labeled every cell type you can think of, I think. So I think I would say you can always get iron particles into cells. Some of the immune cells do require some cationic agent, some transfection agent or something to help out with those. And some cells you're just, you know, never going to get a huge amount of iron in, but I would say we've been labeling T cells, CAR T, Treg, DC, all the types that you might be interested in for things like immunotherapy, and it can be done. We also use things like magnets to help label.
How does that work?
By doing it all on a magnet, on a strong magnet, just to draw iron down, because some of these cells are either a combination of adherent and non-adherent, or some of them are just non-adherent. And if you can get them labeled and then draw them down, then you can enhance their labeling.
Oh, cool.
Yeah. So, and there are a few other ways. If you're doing it, I can definitely talk to you about all the ways we do these things.
Yeah, no, there's certainly been some interest from some of our colleagues. So I was, yeah,
And it's really important, especially for MPI,
Mhm.
what to do after. So we are now routinely doing a magnetic separation and a FICOL on the labeled cells, because if you have any extracellular iron, then your quantitation is off. Right? So it's really important how you treat the cells after they're labeled too.
And in terms of actually, and doing these studies in people, what's, I think Aura already mentioned this, but what is this status of this? Because I guess this is going to be, actually I have no idea what the regulatory path is for something like this.
For MPI?
Or for iron oxide detection by MR, either one.
Yeah, well, there's a long history, right? The iron MRI part went south at one point, and then fluorine agents kind of took over because there was a perfluorocarbon that was approved by the FDA and Health Canada and so on. And we were doing fluorine 19 based MRI, which I did a lot of, and I'm still doing, but in North America, that company just shut down. So that's gone.
Delphin?
Yes.
Oh, okay.
Yeah. They just closed shop. So I don't know, if someone else is going to come back with something different. There certainly are other agents available, not clinically approved in North America. So that could be on hold for a little bit. The ferrium oxetol is being used, like I said, and it's a bit hard to predict because unfortunately, every time I've gotten so far and are ready to do something in humans, there's been interruptions. And so I don't even know how long it takes really either, but yeah, it's hard to answer.
Great.
Okay, so I'll see you next week.
Thank you so much again, Dr. Foster, for this great talk, and thank you everyone for joining. Have a great day and see you next time.